Okay, so what we're going to cover today is kind of what we covered the first lesson of Unit 1, except now we're dealing with the basic unit of living organisms instead of the basic unit of matter. Okay, last time it was talking about the atom, that's the basic unit of matter. Now we're talking about cells. Okay, so if something is a living organism, what are some things it should be able to do or should possess? Like, okay, should be able to reproduce. Okay, what else? Okay, so exchange gases, certainly, yeah, because not all things actually physically breathe, right? Eat, okay, fuel, be fueled, burn that fuel, right? Okay, be able to consume all of that. What's that? Yes, it is. Produce waste, okay? These are all things that a living organism should be able to do. So fire is alive? Well, you just told me that it should consume fuel, exchange gases, reproduce, okay? Fire does that. Sure it does. If a spark from one fire lands somewhere else, you got more fire. You got a whole new fire. That's reproduce. There's no cells in fire. Okay? Fire is not a living organism because it doesn't have the basic unit of all living organisms, cells. Okay? Now, here's where that gets a little bit strange, though. Viruses don't have cells. Yeah? But some people say they're also a living organism. Okay? I mean, they make you sick, right? They get into your bloodstream. But there's a couple of reasons why viruses are debatable as to whether they're actually a living organism. The first part, they don't have cells. The second part, they don't carry out any metabolism. That is, they do not consume food. They do not break down and use that food for energy. And they cannot reproduce on their own. Okay? They use your own cell's hardware to reproduce. Basically, when a virus gets into your cell, it injects its short strand of DNA, okay, into your cell's nucleus. And that goes into the nucleus, and it writes itself in, essentially cuts your DNA and kind of splices its own right into the middle. Right? Now, when that happens, your cell goes around looking for, say it needs to make something like insulin, for example. Okay? There's a section of your DNA that is the instructions for making insulin, and it's something that your body needs all the time. Right? So your body goes in, or your cell goes in and looks for the section of DNA that codes for insulin and says, okay, let's make some of this stuff. Except that that's no longer there. The virus has inserted its DNA into that section. So now your cell, instead of making what it needed, makes a virus. Okay? The virus didn't reproduce itself, it made your body do it for it. Right? So it starts producing a virus. Here's the problem. The cell didn't get what it wanted. So what does it do? Signals again. It says, oh, hey, come on, I ordered insulin. Where's my order? Okay? It's like an angry man at a deli. Okay? Like, where's my soup? Where's my soup? I didn't get my soup. Okay? And it just keeps ordering it and ordering it and ordering it. Well, what does the cell keep doing? The same thing, making more and more viruses until the cell is completely full of viruses and it pops. Okay? And then all the viruses get out and they go find other cells to latch onto. This is why, guys, you've probably all had this experience, right? You go to bed and you got like the little tickle in your throat, and then you wake up the next morning and you feel like you'd have to get better to die. Okay? Right? Because it can happen that quickly. Overnight, your body could produce millions of viruses by that method. Right? Now, how does your body kill viruses? No. Actually, it doesn't. Because viruses are incredibly small. To find an individual virus floating around in your bloodstream or your body would be next to impossible. No, not really. What your body does is it finds the infected cells. It finds the cells that are making more viruses and it kills them because that's quicker and more effective. If you can destroy the things that are making the viruses on their own will die. Because a virus can't carry out any metabolism, has no cellular structures. A virus dies on its own in under a minute if it doesn't find another cell to latch onto. It doesn't have anything to keep it going. So it's smart for your body to kill its own cells in order to kill the viruses. But of course, how does that make you feel? Weak, right? Sick, weak, because your body's destroying itself. Okay? In order to save itself, it's destroying itself. It's like, hey, you look infected. Well, you're going to have to take one for the team. Here you go. Okay? And yeah, that's how it does it. Okay? So yeah, it doesn't kill the individual viruses. That would take too long. Okay? All right. So point it today. We need to know the points of the cell theory. and how they relate to living organisms. There's only 3 points to the cell theory. Okay? We need to know who contributed to the development of the cell theory. So there's a few people, less than there was last unit in the atom. Okay? And recognize the implication of the similarities between plant cells and animal cells. I know when you see them side by side, they look a lot different, but they have way more in common than they have differences. Okay? And we'll look at that as well. All right. First point of the cell theory is what I illustrated to you with that fire example. Okay? The first point of the cell theory is that all organisms are made up of cells. Okay? Some are unicellular, like a paramecium, okay, or like silicates, okay, and things like that. Okay, these are single-celled organisms that secrete a silicate shell, kind of, it's almost like glass, okay, around themselves, and they just have little protrusions of the cell going outside little holes, and they feed by having stuff stick to those, and then they bring it in. Okay, or they could be multicellular, like an animal or a plant, like this cactus. Okay, the key thing here is that all living organisms are made of cells. Okay. This is probably the most important point of the cell theory. What do cells do? Cells carry out the basic functions of the organism. Okay? That means they exchange gases. They break down food for energy. Okay? They carry out photosynthesis if you're talking about a plant. Okay? They transport materials. They can produce materials. Okay? Anything that your body makes is produced at a cellular level. Hormones, things like that. It's produced within the organelles, that's the parts of the cell. Okay? You just have lots of cells doing it, so you can produce large amounts. Right? Now, does that mean every cell does every job? Nope. Okay? Not in a multicellular organism like our cells. Some cells are specialized to do certain jobs. They may not even do other jobs anymore. They become so specialized to do other ones. Okay? Which is why there's interdependence, right? If you take out your liver, you're pretty much screwed. Okay? There aren't many other cells in your body that can do the job of detoxifying your blood as well as the specialized liver cells could. Okay? Or if you were to, let's say, take out your pancreas. Okay? Well, your pancreas produces digestive enzymes that are important for the digestion process. Okay? It also produces hormones, okay, that are important for the digestive process. Insulin, for example, comes from your pancreas. So you have cells in there that are specialized to do certain jobs. Okay? So we have to keep them. Okay, and then lastly, new cells are not created. They come from the division of existing cells. You don't have a factory anywhere in your body that just pumps out different kinds of cells. Oh, it seems like we're a little low on blood. We'll just make a few more blood cells. Okay. Oh, it seems like we want to grow a little bit. Let's make some new bone cells and ship them off to the bones. It doesn't work that way. Okay. Every type of cell in your body comes from the division of other cells that are like it. So when you're growing, okay, your muscle cells are dividing, your bone cells are dividing and making your bones longer and your muscles bigger. Okay? If you are, let's say, you go and give blood, for example. Okay? If you go and give blood, well, then your body's got to replenish all the cells that you gave. Okay? So the blood cells divide. Okay? Now, your bone marrow does a bit of that as well. Your bone marrow can produce some of your blood cells, especially white blood cells. Okay? But they come from the division of stem cells in that case. Stem cells can produce other kinds of cells. All right, so they always come from that division. And that division process is complex. Okay? It's not just as simple as, oh, I'll just split in half and everything will be hunky-dory. Okay? You can't do that. You have to make sure that each, what we call daughter cell, okay, has everything that a cell would require in order to survive. And the most important thing is all of the DNA. Okay? Every cell in your body, with the exception of red blood cells, has an exact copy of all of your DNA. So if I had a cloning machine, I would only need one cell from your body to make an exact copy of you. Okay? Yeah. As long as I didn't use a red blood cell, okay? Because a red blood cell wouldn't have that. But if I used any other cell in your body, it would have all of the DNA necessary to make you up. So that's the first thing that's got to get copied before a cell can divide. So it copies all the DNA and makes sure all the DNA goes to both sides. Then the other stuff, if we don't get the right number of mitochondria or lysosomes or whatever on either side, that isn't as big a deal. The DNA tells us how we can make more of those. But we have to have all of the DNA on both sides. And if that doesn't happen, those cells die. Okay? And if that happens early on during the process that forms, let's say, like an unborn child, okay, then that child may end up with certain genetic anomalies. Okay, like for example, Down syndrome. Okay? Down syndrome happens if either the sperm or the egg, when it was formed, didn't separate the DNA properly. Okay? What happens is an extra copy of chromosome 21 ends up in one of the cells. Okay? And then every cell that comes from that cell has an extra copy, and that leads to Down syndrome. Okay? There's lots of genetic disorders that are caused by the presence of extra genetic material. Okay? Extra genetic material is sometimes survivable. A lack of genetic material is almost never survivable. Okay? So that's what often causes things like miscarriages and stuff like that. Okay? Is that either the egg or the sperm, whichever, okay, had some sort of genetic error in it. Okay? Everybody follow me there? Now, we've also said, okay, every cell comes from the division of another cell. So that means all the cells that you have now, trillions of them, came from one cell. Right? When sperm met egg, okay, it made something called a zygote, which is the single cell that you came from. It divided over and over and over again to form you. Okay? During that division process, obviously some specialization occurred, okay? But all the genetic material necessary to make you was present at that instant. Okay? The instant of conception, everything that had all the potential for you was there. Okay? That's why we as Catholics believe that abortion is wrong. Okay? Because at that point, you were there. Okay? All the genetic material necessary to make you was there at the moment of conception. Okay? Everybody follow me on that one. Okay? That's the reasoning behind that. Okay? Okay. So development of the cell theory. Some important people. Obviously, discovering cells was kind of important in the development of the cell theory. Ironically, this was completely by accident. A guy named Hooke, okay, built the first microscope. And I'm sure that he rolls over in his grave every time he's remembered for being the guy that discovered cells, because he was actually a physicist. Okay? He only built the microscope as a lens experiment on what would happen if he changed the path of light. Okay? That was how he built the microscope. He made curved pieces of glass, okay, essentially to prove that light can travel in straight lines until it meets something that slows it down, and that can change its direction. Okay? That's what he was working on. Okay? And what he found was in a microscope, okay, he had a sample. In this case, it was a piece of cork. So I don't know, maybe he'd had a party the night before and had a lot of wine corks sitting around or something, but he sliced it really, really thin, okay, and he put it on a slide, and he allowed light to pass through it. Okay? So light passed through this specimen, and it hit a lens, which is a curved piece of glass. Right? And as it went through that lens, its path was changed, okay, like this. If it hits straight on, its path really doesn't change very much. Okay, kind of goes straight through. Okay, this stuff here, because it hit at more of a curved edge, was passed over here. Now, look how far apart the four lines were down here at the specimen. How far apart are they up here? Quite far apart. That's how magnification occurs. You change the path of the light, you bend it, okay, and you end up with a larger image. Now, here's the other thing to keep in mind, though. The light from this side of the specimen ended up over here. The light from this side of the specimen ended up over here. Which means not only is it bigger, it's also going to be backwards, flipped. Keep that in mind. It's going to be important for our lab that we're going to do on Thursday. Okay? So keep that one in mind. That's going to be something that'll be important, how a lens works. So Hooke did this, okay? He had this piece of cork sliced up, and when he put it under the microscope, it looked like this. And he saw all these empty little rooms, okay? Like cells in a prison. That's where cells came from, empty little rooms, okay? Why did they look empty? Because the cork he was using was dead. So all the stuff that was inside the cells had decomposed, it was gone. All that was left behind were the cell walls, because they don't decompose as quickly. So what he saw were rows upon row of cell walls, okay, which looked like empty little rooms or cells. Okay, that's where the name came from. So he was able to discover that with a microscope that only magnified 30 times. A good magnifying glass can do that, okay, nowadays. All right, so Hooke discovered cells. That was his contribution. Okay, and I'm sure, like I said, being a physicist, that's not how he wanted to be remembered. Okay, he has a really important law in physics about the elasticity of objects that we talk about in Physics 20 called Hooke's Law, okay, which I'm sure he's much more proud of because it's physics and it's cool. Okay, all right. Then this guy came along during the age of shave your head and wear a wig. Okay, Anton von Leeuwenhoek, okay, was his name. Leeuwenhoek. Leeuwenhoek. Huh? Yes, everything ends in hook. Captain. Yeah. So, Leeuwenhoek made a microscope that looks like this. Thankfully, that design did not catch on because it's very difficult to use. Okay? It's like a ping pong paddle with a little lens in it. Yeah. So what he would do is he understood what Hooke had done with the deflection of light, and he understood that the further that light traveled, the bigger the image would get. So he said, I'm going to stay in a kind of like a dark room, and I'm going to have one source of light. I'm going to put my specimen right here on the tip of this. So if I blew that part up, it would look like this. Okay, so this is where the specimen would go in some fluid, kind of in this top part, and then light would travel through that, okay, and then through the lens, okay, which would make it bigger. And he would put it on a screen, kind of on the other side of the room. Okay, not a projector. Okay, he had only one light source in a very dark room. All right, and what essentially would happen is the image would get blown up. Now that only worked if you could hold it in the right place and then look over your shoulder and look at the image. So it wasn't a great design. Okay, it allowed him to see some stuff people had never seen before. Okay, because if he got it right and the wall was far enough away, things were a lot bigger, up to 300 times, which is not bad. But it's not a design that's easy to use. Right, so we don't really use it. Now, the big thing for Leeuwenhoek is he was able to look at living cells. Hooke never looked at anything that was alive. He did his little cork experiment and was happy because the lenses worked the way he predicted. Right? But, okay, Leeuwenhoek was a biologist. He wanted to look at living things. So he looked at microorganisms that he got in pond water, okay, because there's all kinds of stuff swimming around in there that he looked at. Okay, he looked at blood cells, pricked his finger, okay, looked at blood cells, got sperm cells from cattle. I don't know how he did that. Okay. He's brave, apparently. Because, yeah, bulls tend to be kind of mean, and they don't like people around them, and they have big horns. So, yeah. I don't know how he got that, but he did. Okay? And it says here from cattle, but last time I checked, cattle don't make sperm. Bulls do. Bulls are the males. Okay? Yeah, very early on in a calf's life, if it's a boy, it gets those things removed that make those. Yeah, yeah. Branding day is a tough day to be a little baby calf. Yeah, you get marked and you get cut and you get vaccinated and you get it all on the same day, and you get your tags, yeah, in the ears, yeah. Do you guys actually use the plastic tags or use the microchips? Plastic tags. Okay. So, Leeuwenhoek, first guy to look at living cells. It would be what I would write down if I was writing down a short description of what Leeuwenhoek did. He was the first person to look at living cells. Okay, then came along these two guys, two Swedish scientists named Schleiden and Schwann. Okay, Schwann, that's how you pronounce it in Swedish. Okay, now, they had this idea. The idea was that cells are the basic unit of life. Okay? So they developed a hypothesis which they spent years proving. And the hypothesis was like this. If all living organisms are made up of cells, okay, and tissue from various forms of life on Earth are examined under a microscope, then cells of some form will be present in all of the samples. If, and then, just like we do. Okay? So they looked at tissue samples from plants, from animals, from fungus, bacteria, everything. Okay? Every different type of organism they could get their hands on, they took a tissue sample from and looked at it under the microscope, and lo and behold, they found that there was cells, not all identical obviously, but cellular structure in every living organism. Okay? So that confirmed their hypothesis, which then became the cell theory. Okay, so that's like a lot of people, they'll say to somebody, yeah, I don't believe in that. That's just a theory, which is the dumbest thing you can possibly say. If something is a theory, that means it was a hypothesis that has been proven over and over and over again. Okay, like a theory and a law are kind of the same thing. Okay, they're something that has tons and tons of proof behind it. Right? If you want to say something like that, somebody go, well, that's just your hypothesis. Okay? That would be more accurate than saying, well, that's just a theory. Yeah, a theory has a lot of evidence and backing behind it. So if you ever hear somebody say that, they clearly have no understanding of how science works. Okay? Now you do. Don't ever say it again because it's dumb. Okay? All right. So they had pretty good equipment. They could magnify up to 600 times because they had what's called an oil immersion lens, which is a special type of lens. Okay, you put a drop of oil on the slide and you actually press the lens down into the oil, and it can magnify even more by using the refractive properties of the oil to add additional magnification. Okay, so what did Schleiden and Schwann do? This. They proposed the original cell theory. Okay? They found evidence of cells in every living organism. Okay. As our equipment got better, okay, and microscopes became easier to use, a little less expensive, and could magnify better, people started to notice similarities between different kinds of cells. Right? First one they would have looked at would have been plants versus animals. Yes, at an initial glance and under low magnification, they look very different. Okay? But under a high magnification where you can see what's inside, you find that they have many similar parts. Now, would that happen by accident, or would they have had some common ancestor maybe? Yeah, common ancestor. I mean, if you look like your parents, that's not by accident, okay? It's because you share a lot of genetic material with your parents, okay? And so as a result, you look a little bit like them, okay? Everybody follow me there? Yes? Okay, so basically, finding all these similarities and looking at cell structure of organisms we think are related or maybe are only distantly related allows us to draw or figure out these relationships. And essentially what it's developed is kind of the tree of life, if you will. Okay, obviously at this point, we've got, the kind of different kingdoms of life. We've got like our monorins, okay, which would be your bacteria, okay, things like that. You've got your fungus, okay, you got your plants, and you got your animals, okay. But those would have all come from, in the very, very distant past, a common ancestor who had cells, right? It's not by accident that we all use DNA as a genetic material. It's not by accident that all cells have mitochondria. It's not by accident that we all have, smooth ER and rough ER and all of that, okay? There's no way that plants and animals could have converged and had identical structures. You get that because you shared some ancestor that also had them, right? So that's basically what this is talking about here. Allowed us to draw relationships between the organisms, okay? We're looking here at a bone cell, top left, and cells in the stem of a plant, okay? Both of those types of cells have the same job: support the weight of the organism. Is there a similar pattern or layout here? Dense center, right? Okay, kind of built in circles or kind of a honeycomb kind of a pattern around it. Okay, similar thing. Okay, typically, guys, patterns get repeated if they're doing the same job or a similar job. All right, plant cell and animal cell. Similarities and differences. Okay, here's our animal cell. Yeah, okay, it's got kind of an irregular shape, and it's got, a plant cell has got obviously regular shape and a cell wall on the outside. Animal cells don't have a cell wall, okay? But it leaves that initial thing where, well, those don't look anything alike. Except if I look a little bit deeper, they both have this, okay? They both have a nucleus, a nucleolus, the purple dot in the middle of that. They both have this blue stuff around the nucleus called rough ER. They both have this blue stuff here called smooth ER. They both have one of these, a Golgi apparatus. They both have these things called mitochondria, okay? They've both got cytoplasm, the fluid that fills up the cell. Okay, they've got fibers in there that hold the cell together. All of that stuff is in common. And not only do they have structures in common, but they have biochemical processes that are in common. For example, cellular respiration. Okay? That is, how does your cell break down glucose into energy? Okay? Plants and animals do that exactly the same. Right? Plants get their glucose from photosynthesis. We get our glucose from eating other things. But after that, the processes are identical. We break down that glucose into carbon dioxide, water, and release the energy. Okay? That process is identical. The process by which we make hormones is identical. Okay? We make different hormones, but the process is the same. Okay? So there's a lot of similarities there that we wouldn't have noticed until we were able to look at cells at a cellular level, right? Which is why even back as late as, and it's not going to seem late to you guys, but as late as the late 60s, early 70s, okay, there were only two branches of life, plants and animals. And everything fit into those two kingdoms. Animals. We're animals, yeah. Okay? Here's the problem, though. We knew full well that a lot of things in the plant kingdom had cells that didn't look at all alike. For example, fungus. We're in the plant kingdom. Fungi. We're in the plant kingdom. Okay? Well, if you're a plant, what's one thing you're supposed to do? Carry out photosynthesis. Fungi don't do that. Okay? Fungi have a digestive process more similar to us. They break down dead and decaying material. Well, sometimes it's living material if we're talking about athlete's foot, okay, something like that. Okay, but they consume other organisms for food. Plants don't do that. I mean, okay, yeah, you're going to say Venus flytrap here, but Venus flytrap is still primarily photosynthetic. Okay, so even though we knew fungi really didn't fit in this kingdom, we didn't move them. Okay, they stayed in there for a long time. It wasn't until we said, look, the cellular processes of these cells and the structure of these cells is different than these. It needs to be in its own kingdom. Okay? Other things that went in the plant kingdom? Bacteria. Not all bacteria are photosynthetic, but they had cell walls, so they went there. Okay? That was the logic. Okay? Didn't really fit there. Okay? For animals, all of the single-celled organisms that swim around in a pond were in the animal kingdom, even though they're single-celled. And by definition, an animal is multicellular. So there were lots of things that we were able to prove as a result of looking under a microscope that said, we have to change the way we think, which is exactly what happened in the last unit. We did experiments on the atom and said, that model doesn't fit anymore, revise it. Okay? Same thing happened with biology. Okay? We discovered that, hey, these cells really don't look a lot alike. They're doing a lot of different things. They probably shouldn't be in the same group anymore. Okay? So we changed what we thought as a result of an improvement in technology. Okay, microscopy. I'm just going to run across the hall real quick here, guys, and grab a microscope so you can see what I'm talking about. Okay, so a couple of things regarding the microscopes. First off, they're very expensive. Okay, they're about $400 to $500 a piece to replace. Okay, so we do have to make sure that we handle them in the proper fashion, which is when they get put away, they always have the dust cover on. For some of them, this is a big Ziploc freezer bag, but again, some of them it's the actual dust cover that came with them. They tear easily. Okay. Now, the other thing we want to make sure we do, and the person who used this last has done it, okay, is when we put it away, we want the smallest lens to be in position. Okay. You can see that the shortest of the three lenses is in the viewing position, because that's the one we usually start on, and it's just nice if you put it back for the next person. Okay. The second reason we want to do that is if you leave this one in position, someone will assume, without looking, that the short one is in position, and they'll grab the coarse focus knob, this big one, and they'll turn it, and they'll drive the lens through the slide, okay, which will scratch the lens and break the slide, right? Which now means that the microscope is essentially useless. Once you put a scratch, even if it's tiny, in something designed to make things look bigger, you get this giant chasm that you look at every time you look through that lens now, okay? Because it magnifies the scratch, right? So we want to avoid doing that at all costs. Okay, so we always return it, okay, with this lens in position, okay, for the next person. We carry it in a certain fashion, okay? You always carry it with two hands, always two hands. One on the base, where my left hand is, and one here on the arm, okay? That way, if you get hit in the arm on your way there, you're not going to drop it, okay? Because I've often seen people carrying it with only one hand, and then someone comes up behind them and bumps their funny bone. And what's your automatic reaction when someone bumps your funny bone? Release your hand, okay? And I've seen these come crashing to the ground, okay, on more than one occasion, right? So it's not, I know you're thinking, oh, that'll never happen, but it does, okay? So be mindful of that. Also be mindful of the fact that these come with extremely long, flimsy cords, okay? And these long, flimsy cords are supposed to be wrapped around the base. If they're not, they're at the perfect level to be stepped on. And you won't notice that you've stepped on it until you get to about this point, okay, where you run out of cord. Okay? And then you rip the cord out forcefully from here, damaging the microscope, and you probably trip and fall and drop the microscope on top of it. So always make sure that when you're carrying it, that hand that's on the base has got a hold of the cord, right, so that it isn't dragging on the ground where you can step on it. Okay? Now, our microscopes are a bit better than the ones we left you guys at John Paul II, because that's what you have. The ones we left there when we moved up here, they suck. Okay. The ones that we have, you can move the stage, which is where you put your slide, by moving these two gears down here. All right, which is much better than trying to move the slide with your fingers. Right? Because when you push with your fingers, you inevitably push the stage down, and then you lose the focus that you had worked so hard to get. So whenever you want to move the slide around, you just turn these little knobs here, and that'll move the slide. You put the slide in right here. You just slide it in, and then let that go. It clamps the slide in, and then it'll move around. Okay? And it's nice. Okay? The two focusing knobs. Okay? I mentioned the coarse focus knob before. You see when I turn that, it moves the stage a lot. Okay? With very little turning. In fact, I think you can only get maybe not even one full turn on this knob before it's at the top or the bottom of its movement. The one underneath is the fine focus knob, and you can turn that like crazy. Okay, basically never stops, right? But it doesn't move the the stage very much. Okay, you use the fine focus knob when you're on high power for sure. Okay, and probably on low power. That's the medium lens. Okay, as well. You're going to want to use the coarse focus knob when you're on scanning power. Okay. Do not use the coarse focus knob when you're on high power, right? It has a lot of torque, and you can just push the high power slide right through. Now we do have a screw in here that does prevent the stage from moving up completely, but we found that unfortunately the design of the microscope is that if we put that screw in position where it can protect the microscope, it's also in a position where it can prevent you from getting your specimens focused. So we've had to back them off, right? So they don't really protect the slides and lenses anymore because they were preventing us from getting good focus. Okay, so just be mindful of that. They're delicate. Okay, there's a light on there that's powered. Okay, the switchboard is on the back. Okay, so you got the switch right there. Okay, and obviously these need to be plugged in for that. Okay, in case you didn't know that this goes in the things in the little holes in the wall. Okay, it goes in there. Now, the other thing about these is you don't have to pick up the microscope and set it down in another spot for your partner to look in it. Okay, the eyepiece will rotate. Okay, so you can move that around because every time you pick this up and set it down, it'll go out of focus. Okay, it'll go out of focus every time you set it down. So just because it shakes the whole mechanism and it'll drop a little bit. Okay, so just move this around and that should work. Okay, when you look through the lens, actually I think I have it here. Oh, and I even have a picture of the microscope. That's nice. Okay, when you look through the lens, you're going to see this, even if you don't have anything focused yet. What is that? You would not believe how many people I have had bring me like 3 microscopes on the same day. Mr. Critter, there's something wrong with this one. Mr. Critter, there's something wrong with this one. Mr. Critter, there's something wrong with this one. At about the 3rd time, it's like, really? Maybe it's the operator. Maybe it's you, right? And they're like, but Mr. Critter, there's this big black thing in the middle of the field of view. The pointer? Yeah, it's the pointer. It's supposed to be there. it tells you where the middle of the field of view is. But I have so many times that people bring their microscopes to me. There's this big pointing thing in there. Yeah, pointer. That's what it is. Okay, it's supposed to be in there. It's actually a little wire that's just attached to the inside of the eyepiece lens. Okay, so it'll always be there, right? And it's important because when you get something focused, you want to move it so that it's on the end of the pointer. That way, when you move up to a higher magnification, okay, when you move to a higher magnification, the area you can see shrinks, but is enlarged greater. Okay, if I go to high power, I might only see this much. I might not even see the whole specimen anymore. But if I don't have it on the end of the pointer, let's say I didn't move it, I put it out here, I left it there, I won't see it when I move up to the next magnification because my field of view shrinks as the magnification goes up. Does that make sense to everybody? Okay, so it's always important to put it on the end of the pointer before you move up in magnification. All right, I'm never going to ask you to label a microscope, but it's important that you know the terminology, okay, because that's the terms I'm going to use. Okay, so you got your objective lenses. Scanning power is the lowest, low power is in the middle, and high power is the biggest one. Okay, you got your eyepiece lenses, and we'll talk about the rest of it tomorrow. Okay.